and click this button, and now... All right, welcome to our very large lecture on plant reproduction, plant structures, and anatomy. And it's a big one. It's 39 slides, which means you should definitely be taking breaks during this thing. So, first off, let's talk about reproduction in angiosperms, right? So, gymnosperms, we thoroughly went through pollen and wind disbursement of pollen and landing on the ovulate cones and growing the tubes, right? But there's a fair bit more to talk about when it comes to angiosperm reproduction. So, this is just sort of a little slide showing our roadmap, and let's get into pollination. So, first off, pollination as a term simply means pollen transferred to a stigma. In some of those earliest seed plants, pollen was wind transferred. So, as just mentioned with gymnosperms. Wind-blown pollen doesn't travel super far, right? Roughly 100 meters. For those of you who can't visualize metric very well, that's 333 feet. 11 Oscar lengths. My dog Oscar, he's about 3 feet, or 111 Oscar lengths, excuse me. So, if you lined up my dog 111 times, that would be about as far as wind-blown pollen can travel. So, this limits the gene pool. There's an sort of a baked-in limitation on how many different individuals can sexually reproduce when you have a maximum travel distance. So, instead, through the magic of coevolution, 65% of angiosperms have an animal pollinator. So, 65% of angiosperms have an animal pollinator. So again, this is because of coevolution. The flower is sort of an inherently pollinator attracting structure. You can see some extremely primitive type flowers that are adapted to like self-pollination. But other than that, yeah, angiosperms co-evolved with their animal pollinators, and over 65% of those species, right, over hundreds of millions of years, have developed animal pollinators. So, this gives us a big advantage, right? So, big increase in gene pool for our sexual reproduction. And this, in turn, increases dispersal of genes. Increases, suddenly my brain is crapping out on the term for the whole genome. Genome would be the term. Increases genome dispersal. Basically, now that you can go more than 100 meters, now that it's an animal pollinator taken out, half your genes get dispersed even further. So, your gametes, half your genes are your gametes. So, pretty sweet. Since flowers co-evolved with multiple different pollinators, right? More than just bees. Indeed, the earliest animal pollinators were probably beetles, since bees hadn't evolved by the time you see the first evidence of pollen in the fossil record adapted for being carried by animals. So we co-evolved with multiple different pollinators, and that means big diversity in flowers and species. Huge diversity. So that's pretty cool. All right. Moving on. There's some interesting evolutionary pressures behind pollinators. So, for instance, this right here is a hummingbird attracting plant. So there are a couple of interesting things going on with this hummingbird attracting plant. One, it is bright red. So insects tend not to see the red spectrum very well. So red attracts birds. Right, whereas yellow and white flowers attract insects. And one of the reasons why yellow and white flowers attract insects is because those are very light colors that do a good job of reflecting UV off of the flower petals, which is one of the big things that insect pollinators use. They follow the UV patterns on the flowers in order to get attracted to them. Whereas red flowers don't reflect UV that well, which is great because it selects against insect pollinators. Now, why might that be evolutionarily advantageous? In other words, why limit your pollinator pool? And this is an interesting question. And the answer has to do with the energetic needs of your pollinator, right? A hummingbird here has pretty high energetic needs. And so this flower with the hummingbird pollinator cranks out tons of nectar. And even though it's cranking out tons of nectar, it's still not enough to satisfy a hummingbird's energetic needs. So that hummingbird is going to dip its beak into the flower and get pollen all over its beak. And it's going to drink a bunch of nectar, and then it's going to dip off and go to other flowers in order to fully meet its nectar needs. These have high energetic costs, these hummingbirds. And so they're going to need to visit multiple flowers to satisfy their energetic needs from that nectar. So what's fascinating there is that if insect pollinators arrived, they could gorge themselves on the nectar in a single flower and then satisfy their energetic needs completely. And then there would be no motivation to move to other flowers. And thus, they wouldn't carry the pollen over to other flowers. So, because this flower has a hummingbird pollinator and it produces tons of nectar, it will have evolutionary adaptations to select against insect pollinators, a big one being that deep red coloration, which is pretty cool. If you want to attract moths, put out white flowers. That's like especially white petunias. Moths love white petunias. So that's kind of cool. The energetic needs of your co-evolved pollinator can result in selection against other pollinators because everything I just mentioned. So that's pretty sweet. I dig that. All right. Wind pollination. There's still wind-pollinated angiosperms, right? So a lot of grasses, a lot of oaks, definitely cottonwoods. Cottonwoods are fantastic at generating large amounts of pollen. You know that from the cottony puffs, the fluff balls. So, wind-pollinated angiosperms are still common. 35% of angiosperms don't have a pollinator, an animal pollinator. So, animal pollinators by far the probably the biggest, most effective adaptation there. But if there isn't a huge evolutionary pressure to expand the gene pool, grasses and oaks tend to grow in very windy environments, especially grasses, the prairies. So you can get pretty heavy winds helping pollinate further distance than just 100 meters. You know, cottonwoods, those puffy cottonwoods are adaptations to carry that pollen even further. So these sort of wind-limited pollination plants will often have adaptations to help expand from that 100-meter radius so that your pollen grains can reach further. Okay, so that's our sex. Now let's talk about sex with yourself. Okay, so self-pollination occurs frequently in angiosperms that live in very stable environments. And by stable, we mean basically environments that are unchanging. So things that are not subject to much in the way of disasters, things that have pretty specific and stable climates, right? Self-pollination can become a frequent and helpful tactic. One, you're independent of a pollinator. Independence. You're not reliant on an insect pollinator, right? While insect pollinators are very good, well, animal pollinators. While animal pollinators are very good, it's not guaranteed that any given plant will definitely have its pollen distributed to another plant. So we're not reliant on that. And then related to independent from insect pollinators is you have reduced energy associated with that. You can produce a crappy little flower. So you have a reduced flower. You also produce little to no nectar. I'm not going to write little too many words. So a reduced flower, little to no nectar, right? So this means less energy costs associated with reproduction. And that means more energy for growth and survival. So that's pretty cool. And if you are particularly well adapted to your stable environment, then your stable adaptations, well, they remain. Right? If you have a particularly well adapted phenotype, you have a collection of adaptations that make you very effective in this very stable environment. You have uniform offspring for this uniform habitat. So, pretty helpful. Uniform offspring for uniform habitat. So we can compare this to discouraging self-fertilization. So self-pollination is self-fertilization. Outcrossing is sex with others, not sex with yourself. All right. Oftentimes, it's sex with unrelated others, but plants are not as picky about certain levels of inbreeding. So, anyway, outcrossing is sex with other individuals, not sex with yourself. And the big advantage of outcrossing is it promotes diversity, right? You can get larger diversity of phenotypes, more genetic diversity is shorthand for better adaptability. So more genetic diversity basically means more grist for the mill of evolution, more traits that could be neutral, advantageous, or disadvantageous in the environment. So when we talk about plants that can outcross, we can talk about monoecious versus dioecious plants. Monoecious. Dioecious. What is dioecious? Well, basically, two, and ecious, I expect you all already know what the suffix ecious refers to. So I'll just leave it here. No need to explain any further. So what we're actually talking about is flowers. So, hard cut. I was slightly incorrect when I explained monoecious the first time. So I'm going to leave my dioecious bit in there after this. Although if I refer to hermaphrodite monoecious plants, like hermaphrodite flowers, ignore that bit. That's where I screwed up. I crossed my wires. Cut it entirely, and let's talk about what monoecious versus dioecious is. So, monoecious is where you have separately sexed flowers on the same plant. So basically, easiest way to describe this is you have a spike of flowers. Oh yeah, there's some good looking flowers. And some of these flowers are female. There's some stigmas. Good drawings over here. And then on that same stalk of flowers, you'll have the male flowers. So you don't have complete separation of male and female parts, but you do have separation of the reproductive structures in the flowers. Slightly different. So basically, in a monoecious plant, we have, our pollen producing flowers with our female reproductive flowers. They're separate flowers, but they're essentially in the same compound blossom, I guess is what you might call it in this particular example. They're located together. They are not separated on the plant. So a dioecious plant where it has male and female flowers could be simply that the male and female flowers occur in completely separate parts of the plant. I think most, well, not most, but I think a large number of the dioecious plants, which represent, as I'll say later, 6% of all angiosperms, so tiny fraction. But I think a large number of those dioecious plants are completely separately sexed, where you have male plants and female plants. So monoecious versus dioecious. Monoecious is flowers, so you have the flowers separated by sex. In same, let's just call it physiological location, spot on the plant. Wow, that is getting hard to read. So the same physiological location, the same spot on the plant, the same spike of flowers. A dioecious plant has flowers with separate sexes. So separation of sexes, male and female parts. Now, this can be as simple as having on the same plant flowers with only anthers, and then flowers with only stigma. I should have said stamen. Anyway, so this would be your male flower, and this would be your female flower. So we have a stigma, and we have a stamen. So that's a way to be dioecious. And sometimes you can have whole plants, male and female plants. Cottonwoods are dioecious in that some cottonwoods only produce male flowers and some cottonwoods only produce female flowers. So those cottonwoods cranking out all that cottony sperm, they are male cottonwoods, and they do not have the female reproductive structures on them. Only the female cottonwoods got that. So both of those are dioecious, having separate male and female flowers versus having completely separate sexes of plants. So dioecious is rare. 6% of flowering plants are dioecious. So our big example here is monoecious flowers. So here's a spike of flowers. So pretty cool. Here we have our female flowers on the lower end of this spike, and the male flowers on the top end of the spike, which is pretty cool. So we have flowers on the same spike. It's still part of the same blossoming structure. All the flowers are in the same spot, but they are separately sexed flowers. So the cool thing about these is to promote outcrossing. In other words, to prevent self-pollination, we have kind of two fascinating mechanisms. One, you can have separation, outcrossing by pollinator behavior. What does that mean? Insects that pollinate, so the bees that pollinate this particular flower. So this is, what plant is this? Oh, I did not put down which plant this was. But so this is a monoecious plant. The pollinator actually works in a spiral down the flower stalk. And so they land at the base of the flower stalk, where they encounter the female reproductive flowers. And then they spiral their way up the stalk, right? And then they encounter the male reproductive flowers up top. And that's where they get the pollen all over them. And then the next plant they go visit will, fingers crossed, be, a separate individual. And they do the same thing. They crawl up the female reproductive flowers on the lower end and distribute the pollen from the previous plant. So this is pollinator behavior. So coevolution to promote outcrossing with pollinator behavior. So this big spike of flowers and the bees working in a spiral up them where they reach the female flowers first, then pollen, then move to the next flower and hit the female flowers of that plant, and then pollen and ad infinitum. So that's pretty cool. So another way you could have these monoecious plants have outcrossing is by being dichogamous. Dichogamy is basically the pistil, the female reproductive structure, the carpel / the pistil. I keep talking about, the stigma, but that's just the part at the very tip where pollen lands. So if you'll recall from a previous lecture, this whole thing is a carpel or a pistil. And we have at the top the stigma, and then at the bottom the ovary. So the whole thing is our reproductive structure. Okay, so the pistil matures at a different time than the stamen. In other words, when the pistil is receptive to pollen, the stamens aren't able to produce pollen. And so you don't have to worry about self-pollination because you literally can't self-pollinate because they're mature at different times. So this particular example, these flowers here also take advantage of dichogamy. Fireweed, that's what this is. So this fireweed plant is both taking advantage of pollinator behavior and dichogamy. So those staminate and pistillate flowers, so staminate, so you see there's no stigma in there, so there's no female reproductive structure. And pistillate, big old female reproductive structure. So the staminate and pistillate flowers are fertile at different times. So this even further promotes outcrossing. So that's pretty cool. And then another very cool one has to do with the immune system of a flower sort of being co-opted to promote outcrossing. So this is a molecular pathway system. So pollen tubes are kind of invasive, and there is no guarantee that any given flower will receive pollen only from its species. So how does a flower prevent itself from being riddled with pollen tubes, right, from other species? How does it prevent other species' pollen from building a pollen tube and then ejecting a nucleus into the ovule that is genetically incompatible, right? How do we prevent that whole thing? And the answer is kind of cool. So there are basically molecular pathways to recognize foreign pollen. So molecular pathways detect foreign pollen and they can block or degrade pollen tubes. So they detect foreign pollen and then we release enzymes that block and degrade pollen tubes. Breakdown. So this is an immune mechanism that also prevents basically hybridization events that are destined to fail, right? So molecular pathways detect foreign pollen and then cause a release of enzymes at the stigma that block or break down pollen tubes. But this was a section on how we promote outcrossing and prevent self-fertilization. Well, some plants have this molecular pathway with a sort of detour on it, so they can still recognize foreign pollen, but they also detect self-pollen and cause the same release. So this is incompatibility with self. Incompatible with own pollen. Basically, the self triggers the pollen immune response. Your own pollen triggers this whole response. So that's pretty cool. So that's a pretty hardcore prevention of self-pollination there. When you've got a molecular pathway that detects your own pollen and then releases enzymes that block and break down its pollen tubes, you have a pretty hardcore guarantee prevention of self-pollination. So that's kind of like here we have pollinator behavior and dichogamy, and those are pretty good. But then we have genetic incompatibility with your own pollen through an immune pathway that just nukes those pollen tubes. So that's a pretty effective one. It's probably more effective than pollinator behavior and dichogamy. So anyway, that's pretty darn cute. Okay, now let's go on into asexual reproduction. We've hit sex with yourself, and we've hit sex with others. Now let's talk about reproduction without sex. Okay, so first off, we have vegetative reproduction. So a big part of this that defines it is we're not producing seeds here. Generally, we're producing plantlets or even new plants from parental chunks. So plantlets are basically an itty bitty plant growing on the parent plant that can fall off and then grow a whole new plant. Or you can have like reproduction from busted leaves and such like that. So anyway, I'd probably parts. New parts are cloned from the parts of the adults. So that's a plantlet situation. New plant cloned on a part of the adult plant. What part? That depends on the species. Right? Spider plants, they have a runner that comes out from the plant, and then a new plant is cloned off of the runner. So strawberries also do that. Whereas we have this fun little plant called a maternity plant, also referred to as the mother of thousands, where it clones plantlets on its own leaves. And so it can drop these plantlets off of its leaves, which is pretty cool. So you clone new plants on a part of the adult. And as I mentioned, runners are a highly effective way of getting dispersal while still doing vegetative reproduction. So runners, you get a stem modification that basically grows along the ground and kicks off plantlets. And it produces plantlets. Think strawberries and spider plants. So that's runners. They are still part of vegetative reproduction, cloning that doesn't involve seeds. So all this emphasis on no seeds might give you a clue on the next type of cloning that makes it separate, and that's apomixis. Apomixis is a subset of parthenogenesis. And basically, parthenogenesis is where you still produce gametes that fertilize, but you're essentially producing fertile gametes. So let's do this. You produce fertile gametes that are unneeded. Like there's no true fertilization. I think I confused myself there. Produce fertile gametes. So what does that look like? You know, basically you clone out embryos. If I say fertile gametes, in the case of apomixis, that's good old-fashioned embryos, right? So what that means is apomixis is cloning, but with seeds. Exclamation point. So that's pretty cool. Dandelions are big on apomictic reproduction. The big advantage of apomixis here, you're not getting any advantages in genetic diversity over vegetative cloning, right? There's nothing particularly helpful here. It's still just producing an embryo that's identical to the parent. This is different from parthenogenesis in vertebrates like snakes, Komodo dragons, whiptail lizards, that sort of thing. This is a fundamentally different type of parthenogenesis. This one is true cloning. You're getting identical offspring. But what's the advantage of seeds? Well, in angiosperms and gymnosperms, but this is all about angiosperms, seeds are the mechanism of dispersal. In vegetative cloning, like your best method of dispersal is a runner, and that can really only go so far. You know, here we're able to produce seeds, and seeds can have adaptations for dispersal. See dandelion poof balls. That is the proper botanical scientific terminology for the dandelion reproductive or seed dispersing structure. Poof balls. So, anyway, but yeah, dandelions can literally clone themselves and produce an embryo that produces a seed, and you still get the poof ball that does a highly effective job of catching the wind. So, pretty cool. Okay, so that's reproduction. Feel free to take a break here if you need to. So it's about, I think, 35 minutes, 40 minutes in on the lecture. So good time to take a break. And it's a big obvious slide you can scan to on the YouTubes to get back to. So I'll just take a second to make this part a little longer. So if you take a break, you can come back and easily find this slide so it doesn't just go by in an instant as you're dragging that cursor around. Okay, that's good enough. All right, now let's talk about physiology and anatomy. So, anatomy and physiology of plants. So, we're going to talk about plant tissues, the vascular structures, and specialized structures some plants produce based on their vascular structures. So, pretty exciting. First off is the three types of tissues in plants, starting with the outermost tissue, the epidermal tissue. So, the epidermal tissue is the tissue, the cells that cover the outermost layer of the plant, the cells exposed to the outside. Right? So, the epidermal cells cover the outside of the plant. Main function, protection, and regulating water loss. Right, how do they do that? Well, the epidermal cells are where we find the modifications that allow for gas exchange in a plant. And it was a million years ago, but the stomata. Right, so the stomata is where we have a little structure that is open to the outside. Right? And it leads into the interior of the plant. Drawn this whole sucker out. Very similar to the drawing above, but now I get to deface it. Okay, so we can have gas exchange across the stomata, right? So we need CO2 in and we need oxygen out. And so these stomata have the ability to open a little opening, a little cavity to allow carbon dioxide in and oxygen out. And they also have the ability to shut. So these cells can completely close off the stomatal opening, and now there is no opening, and we have no gas exchange, which sucks because we need carbon dioxide to power photosynthesis, right? CO2 plus H2O yields C6H12O6 plus O2, right? So carbon dioxide needs to come in to power photosynthesis. So why even have these able to close? Regulation of water loss. Because when that stomata is open, one other thing gets out. And that's water. Right? So, when your stomata are open, you have water loss occurring. And that's not great. You might have heard that plants require water. And that is indeed true. So, whatever, I forgot to change the color. Anyway, so when the stomata are open, we get gas exchange for photosynthesis, get rid of that waste oxygen, bring in our necessary CO2. But also when they're open, we lose water. Evaporation takes out our water. H2O evaporates out of the stomata. So those stomata can close in order to limit water loss. So they're constantly playing a game of open and close to regulate how much CO or how much photosynthesis they can do to stay alive and grow versus how much water they can lose before they, get bad. So, guard cells are those outermost layer of cells, and that's where we find the stomata that regulate water loss. Guard cells will often have adaptations that help with protection. So not only are they the outermost layer with the thickened cuticle, right, covering them, so they also can have special adaptations. So you can have, so here, this is a plant hair, which we'll go into later. And these trichomes can often have adaptations to protect the plant. So, protection by creating a tough outer layer covered by a cuticle, and regulation of water through that stomata. The epidermal cells are often, most epidermal cells don't have chloroplasts. However, that's not true for all of them. Many guard cells, the outermost epidermal cells, many guard cells are photosynthetic. which means, of course, that they have chloroplasts. So, most epidermal cells lack chloroplasts. Many guard cells are photosynthetic. So that's kind of cool. Guard cells are the cells around the stomata. And so that's pretty cool. So these specifically, I feel like I was unclear. They guard against water loss. So sometimes they can, yeah, so just to sum that up, in case that left you a little confused, guard cells are the cells specifically around the stomata, flanking the stomata. They are the cells that open and close the stomata, right? Guard cells, many guard cells are photosynthetic. I mean, they're getting direct access to the CO2 right there, might as well be. The CO2, once it comes in through the stomata, then diffuses through the plants. So, I mean, you're right there. So, most epidermal tissues are non-photosynthetic. So, you got the cuticle covering them, and then they have various other structures that may be present. So, and that's not to say guard cells never have like trichomes, but guard cells are definitionally the cells that make up the stomatal opening. So, guard cells are photosynthetic simply because they're right there. Why not, man? The CO2 is right there. The rest of these cells that make up the epidermis, they are non-photosynthetic. So the epidermis that is not the guard cells is non-photosynthetic. They lack chloroplasts. The bonus there is that they transmit light well. So light passes through those cells pretty effectively to the photosynthetic tissue below. So, guard cells are the epidermal tissue that make up the stomatal opening. Epidermis in general are the cells exposed to the outside. Guard cells make up the stomata and they regulate water loss. To a lesser degree, the whole epidermis regulates water loss just by being a barrier. But the guard cells specifically make up that stomata that has to do opening and closing for gas exchange and managing water loss. Epidermal tissue can have adaptations for protection, specialized adaptations, often in the form of those trichomes. We'll talk about it. And then also function as a barrier to the outside, and they lack photosynthetic pigments so that light can get through to the tissue beneath. So, the epidermal tissue is the outermost tissue. Part of our tissue, right, is our trichomes. Trichomes are hairs. So they, at their most basic, they create a little microclimate. What does that mean? That means a beneficial microclimate. They trap cool air against the plant. right? They insulate cool air against the plant, against the stem. This means the plant's temperature is regulated a bit, so it helps regulate temperature. And cooler air has lower rates of evaporation, so it helps regulate evaporation. So that's pretty cool. A lot of trichomes can have adaptations for defense or even offense. So there are trichomes that can secrete sticky substances that trap generally small herbivores. So basically insects and smaller insects, etc. And this can help prevent herbivory, reduces herbivory. So basically a defense against herbivory. I like plants, so tomatoes. The trichomes of tomatoes secrete a sort of glue that sticks those insect herbivores to the trichomes. So it's a pretty effective defense. I, however, love a plant that chooses violence. So, in my little specialized garden, I have sundews. So, no idea how well the webcam will pick this up, but this is my sundew, and the bright red bits off of the leaves are trichomes, and they are producing an extremely sticky substance that traps insect herbivores and then digests them. This is a carnivorous plant. So insects land, they get glued onto it by the sundew nectar. It's not true nectar, but by the sundew sticky stuff secreted by its trichomes. And then the trichomes start secreting digestive enzymes. And they digest that poor trapped insect while it's on the leaf. I used to have a fly problem in here. Emphasis on used to. Mwah. Now I have to feed it fish food because I've run out of flies. So that's a pretty sweet adaptation from the trichomes. Tomatoes have defense. My sundew has offense. My sundew chooses violence. Root hairs are very similar. Trichomes are on the stem. Root hairs, I'll give you a second to guess where you can find root hairs. All right, you were wrong. It was the roots, hence the name, the root hairs. So I don't know what you said, but it was stupid. You should feel bad. So the root hairs are on the roots, and they are extensions that are, anatomically similar to the concept of trichomes, except they are much thinner, much more dense, and they do that thing that all organisms need to do, and that's massively increase surface area. So root hairs are little tubular extensions of the epidermis along the roots that are extremely thin, extremely dense, and they massively increase surface area, and that aids in surface area for water absorption and also soluble minerals. So basically just to help the plant absorb the water it needs from the environment. Pretty cool. There is an electron microscope image of trichomes. This is a tomato trichome with a little glandular bulb secreting that glue. And then this is just a decent image of the root hairs on that first root you get when a seed germinates, which is pretty cool. Trichomes. I talked about the tomatoes with their defensive glue and my sundews with their violence. How about a defensive trichome in this brassica? It sure looks, it's hard to see in this picture, but there's a leaf right down here. And when you look at it under the electron microscope, this is that leaf. And you might be tempted to say, well, that's a cactus leaf. But these are trichomes. So this brassica has trichome needles. Cactus spines are not on leaves, right? That wide, flat part of the cactus that you'd be tempted to call a leaf is a modification of the stem. The stem has enlarged and flattened out for photosynthesis. The spines on a cactus, those needles, those are the leaves of the cactus. So here we have the leaf and trichome spines for defense. Whereas a cactus, you have a stem and leaf spines for defense. So what do we call this one, folks, when you have a similar evolutionary adaptation that occurs in two very different lineages? When two lineages converge on a solution, on an adaptation, convergent evolution. Which is pretty cool. They have a similar appearance, a wide, flattened photosynthetic surface with spines on it for defense, and they grow in similar crappy environments. But in the case of this brassica, it's a leaf with trichome spines, and in the case of a cactus, it's a modification of the stem with leaf spines. Pretty cool. Okay, so that was the dermal tissue. Now, let's talk about the tissue inside. The ground tissue. We have three different types of ground tissue. A hallmark of the ground tissue is that it participates in photosynthesis. Right? That's not all it does, right? Basically, because it participates in photosynthesis, like this is the most common cell type in plants. But it also makes up the bulk of plant tissue. So we have a crap load of photosynthetic cells, but we also have other types of cells. So hallmarks here, they also participate in nutrient storage. Some cells are just essentially glorified vacuoles. right? So the cell has basically had everything in it ground down to a minimum, and it's most of the cell is a storage vacuole. So that could be nutrient storage. Those storage vacuoles, it's not just nutrients. You could also have defensive chemical storage. So it could store toxins. So that's kind of cool. There are specialized ground tissue for protection, specialized ground tissue for defense, storage of toxins. There's a lot of diversity here. So let's separate out those three different types of cells in the ground tissue. Parenchyma. Parenchyma is the photosynthetic cells, the main photosynthetic cells. That makes them the most common of all the ground tissue, right? So that's pretty cool. Characteristics of parenchyma: they have thin cell walls, thin, flexible cell walls. So they allow the plant to have some elasticity. It stands against wind. You don't want a stiff breeze to break a plant in half. So we have these thin, flexible cell walls. And then some have those storage vacuoles, some for storage. Right? Including food, starch, most commonly, water, right? But also toxins, so defensive chemicals, or, in the case of some carnivorous plants, offensive chemicals. So what else do we have in the parenchyma? Parenchymal cells are the ones that secrete nectar. They are also the ones that secrete sap. So, resin. Some plants, they store latex. So dandelions are your great example. When you break a dandelion stem, you get that milky fluid there. Latex is, yes, latex rubber. And the whole thing is on air contact, it coagulates, kind of like how your scabs coagulate, and it forms a scab, essentially. So a protective covering. So, and then they can also, if they're secreting nectar, resin, latex, they're also secreting waste, metabolic waste. Truly the home shopping network of cells. So, pretty cool. They are the majority of cells in fruit. Fruit have almost entirely parenchyma, just because fruits store a lot of sugars and starch. So there you go. In long-lived plants, parenchyma can be exceedingly long-lived. So, in those trees that can live longer than 100 years, their parenchyma, a lot of their parenchyma are going to live the same lifespan of the tree. A lot of them are not going to die and be replaced. They're just going to chill with the tree So these are potentially extremely long-lived cells, depending on how long-lived the parent plant is. So that's pretty cool. That's all parenchyma. Now it's time for collenchyma. Collenchyma are support cells. Right? So they allow a fair amount of elasticity. So they have proteins that a lot of elastic fibers in there. So they promote elasticity. Or my pin could. Oh no. Gray screen is bad screen. Well, who doesn't love a good intermission? Uh, you know, uh, somewhat caused by a crash, but we were talking about collenchyma. Oh, hey, come back here. The collenchyma are a flexible type of cell that support the plant structure, and organs. They have a lot of elastic fibers that mean that they provide a lot of flexibility / elasticity, allows the plant to break without bending, bend without breaking. So that's pretty cool. If you want a good example of collenchyma, these collenchymal cells can be found in celery, specifically celery strings. So when you grab a piece of celery, that give to it is thanks to the collenchyma. So celery strings are an example of collenchymal tissue. And that brings us to the sclerenchyma. All right, so the sclerenchyma is where we find strength and rigidity. And the key thing providing that strength and rigidity is the protein lignin. So lignin is basically this protein that you can pack into the cell walls and provides a ton of strength and rigidity. So packed into cell walls. And like thoroughly packed. Think of it like your outermost skin cells are just packed full of the protein thing. And what's it? I used to know it. I knew it like 2 weeks ago. Yeah. I'll suddenly remember it in a second. Anyway, your skin cells are just packed full of protein. They're basically dead cells that are packed full of something. Well, here, the cell walls get packed full of lignin, and we say they are lignified cell walls. So, if someone you know is being obstinate and rigid and not listening to reason, you could be like, well, aren't you lignified? And they might think of it as a compliment first. Science insults. Okay. So anyway, this provides a lot of strengthening. Lignified stems often have some browning on them. So if you've seen pothos vines, a lot of time those really thick pothos vines get a lot of browning on the outside. That's an indicator that you're starting to get lignification in the ground tissue. So anyway, so this is especially common in stems, right? And it's also common in certain parts of fruits. So, why does a pear taste kind of gritty? Lignin. So, gritty texture of pears is lignification, lignified cells. And then also pits. So oftentimes when you have a seed inside a pit, so for instance, cherries and peaches, right? When you look at a peach seed, when you go down to the pit of the peach, it's this sort of weird cross-hatchy structure thing. And it's this very strong, pit. But this outer layer here, this outer layer is just lignified. It's like a lignified defensive structure. And you break it open to find the seed underneath. So, yeah. Mm-mm. That's not too terrible. So, the pits, the stony shell in nuts and pits, although sometimes the word pit is not a botanical, like, proper plant anatomy term. So while a cherry pit is a stony like a hard lignified structure that you could break open and find the seed under, and same with a peach pit, there are other pits that are the seed. But anyway, peach pit and cherry pits and the stony shell of nuts. Stuff that you can break, hard structures that you break open to find the seeds. That's probably the best rundown of there. So that's a place where you can find sclerenchyma. So lignified cell walls that provide significant strength, common in thickened stems, common in pears, providing pears that gritty texture, and extremely dense in those stony shells of nuts and the peach and cherry pits. So, all right. On to vascular tissue. Here we've got good old-fashioned xylem and phloem. So, xylem is our water vessels. H2O and dissolved minerals. That's what it carries. It is one way. Water is absorbed at the roots, and it goes all the way to the leaves and the stomata. Roots to stomata. So, there's no, backflow of xylem. That's pretty cool. Xylem vessels are often have thickened walls that provide mechanical support. So thickened vessel walls. Provide mechanical support. Woody tissue is secondary xylem growth. So woody tissue is a modification of xylem. Modified xylem. Essentially, you have your core xylem of your sapling, and then you get a ring layer. Of xylem. And then you get a ring layer of new xylem. Right? We are growing horizontally outward. So this is our center. Our center is primary xylem. And our secondary xylem is growing out from there. And the secondary xylem growth is what gets us our wood, which is pretty cool. Transpiration is the one thing I skipped over. So transpiration is water loss. So transpiration kind of refers in general to the diffusion of water from roots to stomata. So our one-way transport. And it's often part of a greater term called evapotranspiration. Evapotranspiration is the general term for the water cycle in a plant. Because we have, our roots, and it goes all the way out to the leaves, and then the stomata, and then, as I said, we have evaporation. So, traveling from the roots to the leaves to the stomata, that's transpiration. So, transpiration is diffusion of water from the roots to the stomata. And it goes hand in hand with evaporation, water loss at the stomata, hence evapotranspiration. And plants have all sorts of adaptations to try and manage evapotranspiration. Okay, so we compare xylem to phloem. Phloem is for transporting nutrients, so plant-produced nutrients, so the synthesized nutrients, and for transport of hormones. So synthesized nutrients and hormones. And it is bidirectional. And that's because lots of parts of the plant are producing nutrients. So it has to be able to flow back and forth. You have your big tree here. And you're going to need to be able to have nutrients circulating, not just from the leaves back down to the roots, but also from branch to branch and branch to branch. And rather than having 12 million one-way vessels, you just have bidirectional phloem vessels to help efficiently circulate nutrients throughout the plant. So whatever branch the leaf is on, we can circulate nutrients any direction we need. So bidirectional to allow efficient circulation of synthesized nutrients, food, and hormones. Not too terrible. Woody tissue actually has an interesting weakness. If you look at the location of phloem in woody tissue, I'll do blue. It's always at the outer edge of the xylem. And every time you get a ring of growth, the phloem in the previous ring dies back. So, in a tree, no matter how wide it is, the only living phloem is at the outermost edge of the tree. So, suffering damage there actually disrupts phloem vessels. If you want to kill a tree, take a chainsaw and make like a 2-inch cut all the way around the base. Just a girdle cut, and your tree will die. Right? So that's pretty crazy. Phloem is near the surface of the tissue. That's why it's generally not a great idea to peel the bark off of something like an aspen. If you see sort of bark kind of sticking loose and you grab it and you peel it off, you may actually be damaging phloem vessels in the process. So, the bark may be cool, but you could be doing some real damage. Okay, roots. There are two main functions of roots. One is the function that heralds back to before vascular tissue, anchoring the plant. So it anchors the plants in or on their substrate. Right? And then what does it absorb? How did you forget this already? I didn't hear any of you saying anything. This is ridiculous. It's water and soluble minerals. Jeez. All right, so that's not too terrible. Okay, so that's the two big functions of roots. Roots can have modified structures. We'll actually go into all sorts of modified structures, but one of the most common modifications of roots are storage of nutrients. So starch storage. And what is this down here that's a modified root? That's right, a sweet potato. Not a normal potato. Potatoes are modified stems. Sweet potatoes are modified roots. But we'll talk about storage. You may have heard, how cactuses can store water in their roots. Don't drink that. It's often extremely alkaline. And you can find yourself poisoned and dehydrated from drinking the water stored in cactus roots. So, maybe don't. Okay, so that's roots. Now let's talk about stems and the good old-fashioned apical meristem. Apical meristem is a fancy way for saying a site of growth. Roots and shoots. So, the very tip of a root and the buds of a plant. Right? So, basically, an apical meristem can be found in multiple locations. There's obviously the terminal bud, that is an apical meristem, but there are also axillary buds, and that is an apical meristem. Axillary buds become branches. So, an apical meristem is a bulge that will develop into leaves and shoots. So, a bud is a part of the apical meristem that will produce leaves and shoots. And, where applicable, flowers. You know, not all apical meristems make flowers. So there we go. Not too bad. Terminal buds are our up. Axillary buds are our out. Up and out to the sides. Okay, so when we look at stem anatomy, we can identify a few features. A node is the location of the axillary bud and a leaf. So, axillary bud and leaf. And also a branch, if you're a branching plant. So, axillary bud, which, as I said, can turn into branches. So, a branch is also a hallmark of a node. Just not all plants produce branching stems. Okay, so a node is an axillary bud and a leaf. An internode is just a term for the distance between nodes. So the portion of stem between nodes. Betwixt nodes. Then we have some leaf anatomy. The flattened portion of a leaf is called the blade. So that big flattened photosynthetic portion, if you're wondering why we're suddenly talking about leaf anatomy, remember when we talked microphyls and megaphylls, a leaf is just a modified portion of stem specialized for photosynthesis. So leaf anatomy is stem anatomy. So the blade is the flattened portion of the leaf. And then the petiole is the little stalk of the leaf connecting to the node. So, the stalk that goes blade to node. Ooh. And I know that's in another spot in this, but whatever. Anyway, a leaf is referred to as sessile if it does not have a petiole. Sessile, a leaf without a petiole. Sessile means it can't move, right? So, like, a coral is a sessile animal, right? It is anchored into its substrate. It can't move on its own. You might say, well, what part of a plant isn't sessile? Well, in this case, that petiole allows for a lot of resistance to let's say, wind and snow and weight being added onto it, essentially it allows the leaf to bend and move around in different environmental conditions. A sessile leaf is going to have much less flexibility. This over here is a sessile leaf. So, there you go. Axillary bud. As again, I guess it's part of the apical meristem. It's an apical meristem. It's at the nodes. And it produces branches and flowers. Flowers in the plants where the axillary bud produces flowers. So this is all very species specific as far as flowers go. But ta-da! Not too bad. Okay, now we're going to go into some specialized stem anatomy, the modifications we can find of stems. So all of these are stem modifications, and let's hit them up. First off, a bulb. A bulb is a bud and leaves. So a modified, I'm covering up the screen there. Modified bud and leaves. Yeah, it is totally registering my hand. Uh-oh. Don't die on me. Don't die on me. Just come back here. No. Modified bud and leaves. Not too terrible. So that's a stem modification that we refer to as a bulb. Our big example is, of course, the onion. Bulbs often have what we refer to as adventitious roots. So an adventitious root is where we lack a primary singular root. We lack the primary root. No primary root. And so instead we just have multiple small roots branching from the stem. Multiple small roots off the stem. In onions, there's like a little stem disc, or a little disc there that makes up the stem. And then you have that bud right there, and then you have all the leaves. So what you eat on an onion is the leaves. Continuing on, rhizomes. So rhizomes are a modified root or modified stem. I guess I don't need to write that since this is a whole slide of stem modifications. So a rhizome is a modified stem that grows horizontally. Against or under the ground. So ferns do this. We've talked about ferns and their rhizomes. This right here, this is an iris. I'd recognize that. So fun things about these, being a modified stem, our nodes are basically along here. In ferns, the stem doesn't grow completely underground, so it'll look different. The iris is a fully underground rhizome. So each one of these blue bits is a node. And on the internode, we'll find, especially on the rhizomes that are fully underground, we'll find scale leaves, which are leaves, but they're part of the buried rhizome. So they're basically just flattened up against the stem, and they're non-photosynthetic because they're underground. So a scale leaf is literally just the leaf that grows out of the node. So it's a part of the axillary buds on that. And then some axillary buds, of course, open up to the surface where you can produce a leaf. And this whole structure is one leaf. So that's pretty cool. That also makes it so the stalk of the iris flower is also part of the big giant compound leaf, which is kind of cool. The stem of the iris is the underground bulb, which again runs into the fact that the gardening and nutrition community do not use anatomical terminology. So sometimes the bulbs are bulbs, like lilies and tulip bulbs are the modified stems that we refer to as bulbs. Sometimes bulbs are rhizomes. So be aware of that. If I'm talking on a test question and I say bulb, I'm going to mean anatomical terminology. Okay, so that's a rhizome, scale-like leaves. So that's notable on there, that grow out of the nodes, just cover the underground root, and then the fact that we have adventitious roots because we're horizontal. There's no singular primary root, right? So singular primary root, think a plant that's got that one taproot and then all of the roots that come off of the taproot. That's actually the large majority of rooting plants. So, all right. Continuing along, runners and stolons. All right. Runners are somewhat similar to rhizomes, but they are not part, they are a modified stem. It's a modified stem, but it is fully above ground or even hanging. Right? So for plants growing on vertical surfaces, their runners can just be hanging out in the air. So what is a runner or a stolon? Well, it's got very long internodes and it's specialized for vegetative reproduction. So specialized for vegetative cloning. Again, strawberries produce little plantlets along their runner. Not too bad. Tubers! Tubers, like your potato. They are stems, right? So it's got a big swollen portion of the stem that's generally underground, but not always, for carbohydrate storage. Carb storage, right? You can tell that they are stems because the eyes of a potato are buds. So those are axillary buds. And axillary buds come off the stem. So, if you let a potato grow and it starts to grow all those leaves coming off of there, your potato is technically branching. So that's kind of fun. It's a modified, specifically a modified rhizome. But modified stem that is all swole up for carbohydrate storage. The eyes are those axillary buds. And basically, leaves drop off very early in development because oftentimes these things are underground, right? The majority of tubers are going to be underground. When a plant is growing its tubers, some of those might be on the surface and actually manage to leaf out. Most of those tubers are going to be underground, and those leaves will drop off and leave little scales. So, scale leaves again. More modified plants. Tendrils. Tendrils aid in climbing. Climbing and attachment. In grapes, this drawing here is a grape, right? So that's a grape tendril. In pea plants and pumpkins, those tendrils are green. They're not part of the woody stem. They're modified leaves. But guess what? Leaves are modified stems. So tendrils modified from leaves are still modified from stems. Ha! Don't have to change my slide. And then last up are cladophylls. You remember how I told you about how cactuses, what looks like their leaves are actually wide, flattened stems, right? That's what a cladophyll is. So, wide. Well, I do not have enough space to write there anywhere. Cladophyll. Wide, flattened stem. And it's, of course, specialized for photosynthesis. Hence, throwing fill in there. For all that chlorophyll built up in there. That's stem modifications, and that's like 38 minutes. So good time for you to take a break. Just leave this up here for a second. Pretty. Okay. And we're back. Let's talk about leaves and modifications of leaves. This is a long one, man. There's officially 21 slides left. We're at slide 18. Hence why I'm like putting in breaks. Just manually discussing breaks. Okay, so leaves are modified stems. So modified stems for photosynthesis. They are characterized by having a waxy cuticle on the upper layer, the upper surface has the waxy cuticle, and then the underneath, the underside, is where we find all our stomata. So let's talk about modifications of leaves. There's our roadmap, and boom. Okay, so first off, bracts. A bract is a floral leaf. This may sound silly to say, as a flower petal is technically a leaf. But in the case of for instance, poinsettia, which that is, that purple is not coming through. So, poinsettia. Those are not flower petals. Those are just leaves. Floral leaves. If you look at the actual reproductive structures of the plant, they are coming off an apical meristem on their own. In other words, you have, the apical meristem, and then you have, there's some male structures, and then here's my stigma right there. And then down here at an axillary bud, we have our colorful poinsettia leaf. It's a colorful leaf, and here we have no petals. So our reproductive structure is on its own without petals, and the colorful, attracting pollinator stuff is a leaf, which is a bract. So poinsettias are your big example. If you're familiar with dogwood flowers, those are also a bract. So red, white, or pink. Red attracts bird pollinators. White attracts insect pollinators. Pink, probably both. And they have a very similar function to the flower petal, but they're not an actual part of the terminal bud that produced the flower. They're not a part of the bud that produced the flower. They are leaves very close to that. So, ta-da! Okay, fun down here, cactus spines, right? The cladophyll is stem, so our cactus spine is I'm leaning way down there. I have no idea how much of that was me actually being visible. The cactus spine is a leaf. Now, let me remind you that microphylls were only present for one group of seedless vascular plants. From ferns onward, we have megaphylls. So pine needles up here, these are megaphylls. Cactus spines, these are megaphylls. So don't get fooled by that. So there we go. The cactus spines are, of course, a leaf modification for defense. Not too bad. Cactus spines are separate from rose thorns. Rose thorns are direct defensive structures on the stem. Cactus spines are the leaves. Now, being reduced to these defensive spiky bits is great for defense. It's also great for managing water loss. It reduces surface area for water loss. Evapotranspiration. So we lose less water out of the cactus leaf because it has very low surface area. Okay, pine needles. Basically, similar idea. They are reduced surface area. They're actually flattened leaves if you look at pine needles, which is kind of neat. And again, they have a reduced surface area to manage evapotranspiration, to manage water regulation. So that's pretty cool. Another cool thing about pine needles, being these flattened needle structures, is that they don't catch water. So not only do they reduce evapotranspiration by having a reduced surface area, but they don't catch water. So what limited rainfall you get where conifers grow, it goes to the ground. H2O to the ground and the roots. So that's kind of cool, right? Plants don't absorb water through the stomata. At best, a stomata, like water, a big water droplet, like the upper surface where water droplets might collect is a waxy cuticle. Water can't penetrate it. So water left behind on a needle is just lost water. Conifers do really good at super high elevations where there's high rates of evaporation and low rates of precipitation, low rainfall rates, high evaporation rates. You get a lot of these needle plants that have very low surface area to reduce evapotranspiration, and they also allow water to just bloop past the needles to the ground, so they increase water available for that absorption. Is my frog making noise? Frog! Nope. The moment I moved the microphone to catch it. All right. Insectivorous leaves. Leaves that choose violence. What's that? Leaves that choose violence. These leaves are modified to trap insects. They are highly effective in environments where there's extremely low nitrogen in the soil, so extremely nutrient deficient environments. You have a lot of insectivorous plants. So Venus flytrap leaves actually have little spines on them that a bug will move past and trigger the plant to shut. That leaf will reopen, so I didn't deny it a meal. It's got plenty more. The great thing about Venus flytraps is the trap itself can become just basically a photosynthetic leaf. So it produces way more traps necessary than to get the nutrients it needs from its insects. Another cool modified leaf is right here in my pitcher plant. So if you look at the tip of my leaf, there is no separation between leaf and pitcher. So this pitcher has a nectar-like fluid in there that attracts bugs, and they crawl in there, and then the inside surface is extremely smooth, and they fall into the nectar-like fluid that is actually digestive fluid, and they get trapped and they die. More leaves that choose violence. So, I have a pretty cool carnivore garden. If you want to make your own carnivore garden, my recommendation, I'm going to take a second for a break and put these back because they're drying out here. But my recommendation for a carnivore garden is to put it under an extremely bright LED growth lamp, and then only water with DI water, like pure, not spring water, but like the distilled water from the store. And then what you do is you give them insects, very small crickets for my fly trap, or for my my sundews and my pitcher, small amounts of fish food mixed with just enough water to make a paste. And that keeps them going. But I need to put them back in the light and the water. And action. Okay, so that was modified leaves. So now let's talk about plant defenses that aren't just direct violence, but more like, defense against herbivory. So, types of defenses include anatomical defenses. So, what are anatomical defenses? Anatomical defenses include, well, let's go with the classic rose thorns. How about those modified trichomes on that brassica from way back when I was talking about how it's not cactus spines? Or cactus spines. Those are all modifications. Forgot my pen. Those are all modifications for defense. So, rose thorns, trichomes. Remember our tomatoes that produce glue, and the brassica that produce trichomes that are like needles, and then cactus spines. Chemical, speaking of the glue associated with those defensive trichomes. So more like, our brassica spines. Would be a purely anatomical defense. The tomato glue falls into chemical defense. So chemical defense, poisons, irritants, etc. My favorite biological category. So let's talk about poisons. First of all, poisons are generally referred to as secondary metabolites or secondary metabolic compounds. The reason they're referred to as secondary is because primary metabolites, primary metabolic compounds, are the hormones for growth and reproduction. So, just a classification thing. Secondary metabolites that are for defense, in other words, plant poisons, these are expensive. So they are energetically costly. So the cost-benefit ratio better be helpful. You have a high cost when it comes to this benefit. But the benefit is for defense against herbivory. Not getting et. So, general rules about plant poisons is that the toxin generally does not affect the plant. And basically, it's often found in membrane-bound structures, so stored in organelles. That's a fancy way of saying found in membrane-bound structures. I don't know why Past John wrote that. Stored in organelles. And one of the kind of neat things is often they are harmless until metabolized by an herbivore. In other words, they won't kill the plant because they require, digestion in the intestine, right? So metabolized by the herbivores, generally in the intestines. So let's start with some real dramatic plant poisons. How about cyanogenic glycosides? Cyanogenic glycosides. So, cyanogenic glycosides are one of those that are harmless until metabolized. So, inside the plant, cyanogenic glycosides are harmless in the plant, and they are converted to cyanide when digested. That's a pretty hardcore plant defense chemical. Cyanide inhibits electron transport. And if it inhibits the electron transport, then it inhibits, if it inhibits electron transport, then it also stops the electron transport chain. And let me tell you, your cells run through ATP real fast. And if it stops the electron transport chain, it stops ATP production. So cyanide shuts down ATP production and thus shuts down organs real fast because cells run through ATP on hand real fast, right? Recycling ADP back into ATP is a huge survival mechanism for living organisms. There's nothing alive that doesn't do that. So shutting down the electron transport chain shuts down the recycling of ADP plus P into ATP, and it kills the cells. So no more cellular respiration, no more ATP, dead. So the cassava. The cassava is a modified root, and it originates in South America. If I'm remembering my facts correctly. And it has become an important food source in Africa. And the outside, the epidermis of the cassava is rich in cyanogenic glycosides. So you have to prepare that real well. You have to make sure there's not one bit of epidermis on that root left. So basically, you have to scrub that completely. And then after scrubbing it, you probably soak it and throw out the water just to make sure. And I really want to know the cultures who eat cassava what the process was for figuring out the right way to prepare cassava. Because I know in South America, there are some people who just soak their cassava root for a couple days and all the cyanogenic glycosides leach out. So they just drain off the water and just keep soaking it. Where in Africa, they scrub the outer parts of it completely. And I just want to know what the process was for that. Just like watching people die. Like, well, I guess that wasn't it. Well, cooking didn't stop it. Okay, glycosides. Again, cardiac glycosides. So cardiac glycosides are a little different. They inhibit the sodium-potassium pump. The sodium-potassium pump, NAK. If you haven't been to Human Anatomy and Physiology, when we say the brain runs on electricity, your brain sends electrical signals. It's not like electricity from the wall. What it is charges, like your neurons that actually connect together to send the electrical impulses. Basically, there's the charge outside the neuron, and then there's the charge in the cytoplasm of the neuron. So the charge outside the cell membrane and the charge on the inside of the membrane. And when the charge outside and inside become equal, it instigates this chain reaction that causes that charge to become equal all the way down the length of the neuron. And that's the electrical impulse traveling. So it's charged, and it's a charged impulse that travels. So what maintains those charges? The sodium-potassium pump. The resting state of a neuron is the inside is ever so slightly negative compared to the outside. And the sodium-potassium pump pumps 3 positively charged sodium out and 2 positively charged potassium in. So you take 3 positive charges out, 2 in, and you have decreased the charge by a 1 to maintain the resting ion charge difference outside versus inside. You shut that down and you slow and stop nerve signals. It's lethal in higher doses, but everything is toxic in a high enough dose, and everything is non-toxic in a low enough dose. And there is a toxic dose of this that is, not a beneficial level of toxicity, right? If it slows or stops nerve signals, well, a heart attack is when your heart's signals, the nerve signals of the heart, go wild, right? So your heart rate speeds up. So you can treat an arrhythmia. I hate the word rhythm. By slowing those nerve signals. You don't give someone enough cardiac glycosides to kill them. That's sort of against the plan of treating their arrhythmia. I mean, it will stop their arrhythmia, but I think the idea was to not kill them. So there you go. All right. People in Africa, there are people in Africa who use cardiac glycosides on arrows, so they can use them as arrow poison. So that's kind of cool. Those are 2 human uses. Let's talk about the wild biological world. Milkweeds, right? Their cardiac glycosides do a great job of inhibiting herbivory. It's harmful to it's harmful to vertebrates. They eat enough of it and they can die. But it is lethal to insects. Of course, the milkweed flower has a pollinator. So it has multiple insect pollinators, but the milkweed flower's pollinator is the monarch butterfly. And one thing about the monarch butterflies that make them very cool is that they love to eat milkweeds, and then monarch butterflies lay their eggs on milkweed. And their caterpillars have evolved a resistance to cardiac glycosides. So the caterpillars are unaffected when they eat the cardiac glycosides. And what they do is they incorporate it into their skin. And that becomes a defense against predation. And it is such a useful defense against predation that after they spin that cocoon and metamorph into a butterfly, they still have those cardiac glycosides incorporated into their skin, into their tissues under their exoskeleton, which is pretty darn cool. So, if you are a bird and you see a tasty, tasty butterfly that you have caught, a meal, and you find out that you have just eaten a monarch butterfly, you will go, that is bird tongue. That is literally, so that bird has discovered that the cardiac glycosides taste horrible. Good news, it doesn't eat enough butterfly to die. That would actually be counter to your predatory defense, right? Killing one predator sounds pretty effective, but if instead the predator kills the butterfly and then vomits, has a bad time, it won't eat that butterfly ever again. And being birds, that's information that can be relayed amongst birds. So they try it once and never again. So this is the monarch butterfly. And a cool thing, anytime you have a poisonous, an animal that's poisonous, there's almost certainly going to be mimics that don't exert the metabolic cost of incorporating poisons in there and having caterpillars that are resistant to the poison. But they certainly look like the poisonous one. So if this bird has eaten the poisonous one and gone blah, it's less likely to eat the viceroy, which is kind of cool. You'll find the same thing with venomous organisms too. If you have a venomous snake, you'll probably find some non-venomous snakes that have a similar coloration or pattern. So that's kind of cool. All right. Next up is alkaloids. Let's talk about alkaloids. This is a very general category for plant defensive chemicals. So defensive alkaloids. There are large amounts, plant to plant. 10,000 plus different defensive alkaloids out there. Caffeine. Lethal to insects and many vertebrates. Lethal to insects and harmful to vertebrates. They have to have a lot of caffeine to get killed, but harmful to vertebrates. Humans are special. We can imbibe extremely large amounts of caffeine. Nicotine. Again, lethal to insects. Right? Cocaine. Lethal to insects and harmful to vertebrates. Morphine. Lethal to insects and will cause vertebrates to get real sleepy, which actually may make them more susceptible to predation. So if a vertebrate eats a bunch of morphine and eats a bunch of opium poppies, so the opium poppy flower right here produces morphine as a defensive chemical. If you have a vertebrate that comes in and munches them flowers, it will get real high and sleepy, and then it might be easier to get at. So might have multiple mechanisms there. Not only is it harmful, just due to the anesthetic properties of morphine, but you can get to the trip balls levels of morphine and become very susceptible to predation. Speaking of the trip balls mechanism of defense, another thing that's lethal to insects and causes vertebrates to trip is good old-fashioned D-lysergic acid, which you might know as LSD. So, lethal to insects, hallucinogen. To many vertebrates. And again, if you're tripping, you're probably a lot more susceptible to predation. And if you're just eating plants, you're also very likely to eat too much and die too. So it's a bad way to go. So like I said, nicotine on tobacco plants, it's a metabolic disruptor. So for insects, metabolic disruptor, it kills insects. Which makes it all the more interesting that in humans, it's mostly harmless, except it's highly addictive, which is kind of fascinating. An insect defense for the tobacco plant. Addictive chemical for humans. D-lysergic acid is in morning glory flowers, especially primarily the seeds, but in the flowers as well. That's kind of cool. You might also know of D-lysergic acid being in another different group of organisms. What's that? Did you say shrooms? Yeah. Magic mushrooms produce D-lysergic acid. And you know what's fascinating about that is that fungi are much more closely related to animals than plants. So this is another example of when a similar evolutionary solution, a similar evolutionary adaptation develops in very different lineages. Convergent evolution. Morning glories and the fungus among us. So that's pretty cool. Tannins. All right. Tannins can be found in bark. So, they are basically a chemical that is going to interfere with protein digestion. So, found in bark. And they interfere with protein digestion. So they help defend against herbivory, especially from the organisms that like to chew bark. Humans have found out that tannins are quite effective when it comes to treating leather. So that's kind of cool. You can convert raw animal hide to leather, and that's called tanning. Leather is much more resistant to decay and durable than just hides. And then oils, right? Oils are also in plant defense, although in general, oils are strong odors. Their defensive mechanism is in repelling herbivores. So the odor repels herbivores. And weirdly, for a lot of humans, it smells delicious. Peppermint and sage oil, that smells delicious. Man, plant defenses suck against humanity. I don't know what it is about us, but when it comes to plant defenses, a lot of them fail. Thumbs up to the cyanogenic glycosides and the cardiac glycosides. Those still work. Okay, so this is all about defense against herbivory. What about protecting yourself? Okay, so I've got a slide coming up here where I've made a fundamental change to the slide that's uploaded for the student lectures. That's because I recently did a standard check over spring break on slide information and found that the example of allelopathy, which I'll go through in a second, but the example of allelopathy, the black walnut, is a crap example. It's a great example of actually, weirdly, a story going viral in the 1920s through the 40s and having little to no scientific backing on it. So, you'll see some stuff about black walnut trees, and it's all false, fascinatingly enough. Allelopathy is basically when you have plants protecting themselves from other plants, protecting from their competition. So, plants protecting themselves from other plants. They're generally secreted by roots. And essentially, they build up in the soil. And they inhibit competitors. They inhibit growth of competitor plants. That's allelopathy. Basically, minimizes competition. Protects from competitor plants. Secreted by the roots where it builds up in the soil and inhibits the growth of competitor plants. Minimizes competition. There's a little bit. Okay, so why? First off, my example here is a plant called sorghum bicolor. Millet, you might know it by in the agricultural world. It's an important agricultural crop, which is kind of cool. Sorghum bicolor secretes a chemical, sorgiolone, I'm not sure. It secretes it from the roots into the soil, right? And it inhibits the growth of nearby plants. So it gets absorbed by, so it gets secreted by the roots into the soil. And over here in this picture, A, you can barely see it, but here's a really sad seedling here. There's a very sad seedling there. These are competitor plants. Those are little pea plants. And then this is sorghum in its native sandy soil substrate. And here in this one, they have sorghum in its native sandy soil substrate, and then they amended the soil with activated carbon, which breaks down that allelopathic chemical. It breaks down sorghiolone. Man, that sounds like an Italian movie maker who made westerns in the 70s. Anyway, timely reference. So these were planted at the exact same time, the only difference being that they amended the soil in B with activated charcoal, which broke down sorghiolone. And these are the same pea seedlings, except doing really, really well, and there's a lot more of them. So you can see sorghum bicolor is awesome at allelopathy. Okay, so why did I have to ditch a whole slide, a whole example on black walnut? This is a fascinating story, and it gets at the heart of why studying allelopathy really needs to be done by ecologists and not like the laboratory botanists that have done it in the past. So let's talk about how laboratory botanists first studied allelopathy. What they would do is they'd see some plants not growing particularly great around other plants that are growing great. And they'd say, well, I bet that other plant is secreting a chemical. And then they'd take that plant and then they'd grind up leaves, stems, and roots, and then they'd make extracts, and they'd like extract phenolic compounds and other volatile compounds. And then in their greenhouse laboratory, they'd take their extracts and add it into the soil for the plants, and they'd try and grow seedlings in that soil, and they'd record whether or not those seedlings sucked. And sure enough, in black walnut trees, when you grab leaf extracts and extracts from the nuts, you can get a chemical called juglone or yuglone. I don't know. Juglone. Anyway, you take those extracts with juglone in them, and it will sure inhibit the growth of plants, all right. Thing about it is, if you look in the soil under black walnuts, like when you have a trimmed black walnut where a lot of light can get to the area underneath it, things grow fine. Ecologists doing studies find not nearly enough juglone in the soil to actually cause allelopathic interactions with other plants. There's little to no juglone in the soil around black walnuts. Yes, if a black walnut tree were to atomize and rain down its ground up pieces onto the soil around it, plants would be inhibited. So, but as it turns out, things didn't tend to grow under large wild black walnut trees because large wild black walnut trees had very large canopies which blocked out the sunlight. And they did a very good job extracting like nutrients from the roots when they were a very large mature tree. And so like the soil was, nutrient deficient because the black walnut had spent a long time growing there. And there was little light, and so there was very little growth around there. But laboratory scientists said, I bet it's allelopathy. And you find that in so many different examples of allelopathy. It took me tonight 2.5 hours to find sorghum bicolor, which is actual allelopathy, where an ecologist literally planted it and in the soil and then did soil measurements of concentrations of sorghiolone and actually demonstrated an ecological allelopathic effect. So, turns out the black walnut thing, the whole nothing grows under a black walnut, was first published in a few farmers' almanacs in the 1920s. And in 1948, there was a U.S. Army Corps of Engineers little paper that said, hey, nothing grows under black walnuts. And in the 1970s, someone cited a publication about black walnuts causing wilt in plants growing underneath them. The 1948 thing is so old that it didn't even talk about juglone. And it's out of circulation, so it's kind of useless as a source. And B, the 1970 citation doesn't lead to a paper. There's no paper with the title of that citation, and the authors listed didn't have a bulletin paper on black walnuts associated with that. It's a ghost citation, and it turns out one person cited it, and then everybody else took the citation from that one person and just added it into the paper, which is why you find the citations, people. Look it up for yourself to make sure that it's relevant to your paper. Otherwise, you're going to have 50 years of people citing each other in a grand, incestuous, fake, not fake, but ghost citation that didn't actually exist. So, as it turns out, black walnut, juglone, you can find common stuff in which people talk about its allelopathic effects, and it's just not. It's just not allelopathic. It blocks out a lot of sunlight, and an old one, if the soil doesn't get constantly amended with, dead things and stuff like that, the soil can get nutrient deficient from having a big old tree there for a long time. Oh, man. Okay. Apologies. All right. So, that's allelopathy. Another secondary metabolite that's been in the news lately is phytoestrogens. And this is another one I just went down a rabbit hole correcting because, fascinatingly enough, phytoestrogens, especially talking about soy here, are a chemical in plants that have a similar chemical structure to estrogen. Now, similar does not mean is. Phytoestrogens aren't human estrogen. So, they have a similar chemical composition to human estrogen. And they were thought to have an impact. You saw, people freaking out about this stuff, I don't know, 10 years ago or something like that. You can still find the people around going soy boy, soy. Anyway, as it turns out, all that crap is ludicrous. Which you might imagine based on the fact that countries where there's a lot of soy ingestion in their diet don't have all the effects people were saying. But to go through it just real quick. Phytoestrogens in vitro, so in tests where you had human cells in petri dishes, and in animal studies like sheep and rats, were shown to have pretty significant effects. They were pretty significant endocrine disruptors. So, for instance, even back in the 1940s, female sheep grazing on clover-rich pastures. Clovers produce phytoestrogens. They had abnormally high rates of infertility, abortion, and reproductive abnormalities due to the phytoestrogens disrupting normal estrogenic activity. Basically, it disrupted pregnancy. Then when you added the phytoestrogens, the isoflavones, into rat diets, it suppressed female sex behavior in rats. And male rats had increased anxiety and elevated stress. And then monkeys had higher frequencies of intense aggression and submission behaviors, as well as decreased time spent in physical contact with other monkeys, right? So sheep, rats, and monkeys, man, these were having significant effects. Wildly different significant effects. Like, they weren't recording birth defects in rats or monkeys, and the monkeys were correlated behaviors. Like, there's no causal mechanism in here. It's all been hypothetical mechanisms, right? But then you get into like studies where they start to measure hormones. So, for instance, soy-based infant formula, you had phytoestrogen concentrations at 1,000 nanograms per milliliter, which was like 13 to 20,000 times higher than estrogen levels, the endogenous estrogen, and 50 to 100 times higher than the estrogen levels in pregnant women, 3,000 times higher than the estrogen levels at ovulation, right? So this is like a big deal if it had an effect. These were similar blood levels to those that they found in animal studies that produced all those concerning effects in rats and monkeys and sheep. But again, humans are not rats and sheep, and we're not even, monkeys. So cladistically, if you create a group called monkey, then we're in that one. But that doesn't mean we are biologically the same. Anyway, they even found in infants fed on soy diets vaginal epithelia that appeared to be affected by estrogen at times when the breastfed or cow-based formula counterparts did not. But more recent studies indicated that there were no medically significant effects here. Sometimes you could see something estrogenic like that, but it wasn't significant. It meant nothing. Like, oh, wow, what does that mean? No consequence. Oh, okay, then. Like, I can eat lots of stuff that will mess with me and won't be medically significant. There were no differences in growth on infants fed soy formula diets. There were no differences in subsequent development, no differences along the line in puberty. Compelling blood tests and compelling physiological effects at a tissue level do not equal real systemic effects. In 2009, they did a meta-analysis, and they were like, wow, it increases cycle length and like reduces the number of days a woman could be fertile. And then there were studies on how soy diets affect pregnancy, and they found at max the phase at which you're fertile, the follicular phase, might be reduced by an day. And overall, there were no effects on fertility, except that, interestingly enough, people with fertility issues who were receiving fertility treatments and had a high soy diet had higher pregnancy rates than people just getting the fertility treatments alone. So that's fun. There's a lot of correlational stuff that's interesting. There's a lot of blood tests that are like, holy crap, these phytoestrogen levels are through the roof. And then when you do actual longitudinal studies, we have found that the phytoestrogen effects in soy and other plants are nil. Nada. The importance of science. Sometimes a blood test saying 13 to 20,000 times higher circulating levels of phytoestrogens than normal estrogens can sound pretty terrible. But again, you're not a plant. So plant estrogen is not going to do the same thing as estrogen. Okay. Okay. All right. So, fascinating. So, do soy foods stop the stork? No. If you eat a bunch of soy and you drink a bunch of soy milk, are you feminizing yourself? No. Does soy have any medically significant effects in longitudinal studies? No. So, the word phytoestrogen and the word endocrine disruptor certainly sound dramatic, but you gotta really look for this stuff to see what the science actually says. So, now, like I said, there are effects in non-humans. This is kind of a cool ecological interaction here. This is the California quail. It lives in along the Pacific the Pacific coast, all the way through the Pacific Northwest, and even a little bit into the rainforests of the Pacific Northwest. It does really well when there's lots of nice precipitation and like grasses, forbs, and it eats those preferentially. But in drought years, the only thing surviving is desert annuals. And a lot of these desert annuals produce high levels of phytoestrogens. Now, quail are kind of legendarily fecund, which is a real fancy word for they have a lot of babies. Quails produce a ton of babies. And so if the quails are eating up these desert annuals that have sort of a relatively delicate ecosystem here, and they are reproducing at California quail rates, in a drought, you could easily wipe out this stuff. But as it turns out, these desert annual phytoestrogens, they inhibit California quail reproduction. This benefits the desert annuals in that there's not going to be a bunch of baby quail out there multiplying the amount of animals trying to eat enough desert annuals to stay alive, right? So this is a great defense against herbivory for the desert annuals. But what's extra fascinating here is that this is really beneficial to the California quail because having offspring during a drought is unlikely to produce much in the way of successful offspring, and due to parental care, will be a high energetic cost. So when the phytoestrogens that these quail have to eat, during the drought, shut down their reproduction, it saves them energy. So that's kind of a cool ecological interaction there. Not just a defense against herbivory, but an advantage for the herbivore. So I think that's pretty cool. And then you can look at the scaled quail, and you can see it lives in the desert, and its diet is in large part these desert annuals with high concentrations of phytoestrogens, and scaled quail are unaffected. They are adapted against the effect of phytoestrogens. So they have adaptations against the endocrine disruption of phytoestrogens. So that's kind of cool. So they can successfully reproduce while eating these desert annuals. So this is coevolution right here. Although one could argue that there's a bit of coevolution to the California quail and the drought. So kind of cool. All right, continuing on. How about when a plant defense it becomes a liability when you've got humans involved? So this is the Pacific yew, Taxus brevifolia. So the Pacific yew tree, and it produces Pacific yew taxol. And Pacific yew taxol, who wants to guess what it kills? It's lethal to insect predators. It's actually pretty good against vertebrates as well, which is kind of cool. Well, no, actually, it's just insects. I'm misconstruing here. So it's just lethal to insects. But as it happens, it's a spindle inhibitor. And what's kind of fascinating is if you give it to someone with cancer, it slows and inhibits mitosis in cancer cells. It has significantly less effect on non-cancer cells. And it has a low effect on healthy cells. So as it turns out, you could give someone a transfusion of Utaxol in there. This was the first modern chemotherapy drug. And as a byproduct of that, it went nearly extinct. Why did it go nearly extinct? It was the first modern chemotherapy drug, and when we first started using it in chemotherapy, we could not produce it in the lab. So, Taxol we managed to find a laboratory synthesis pathway for Utaxol. So now we can use it in chemotherapy without actually destroying Pacific yew. So Pacific yew is on the mend. Hooray. So it's a significant plant in the Pacific Northwest ecosystem. So it's a good thing we didn't drive it to extinction, but it was such an effective chemotherapy drug that we did nearly drive it to extinction. All right, the cinchona tree, which the species is called the cinchona species. The species is, of course, named by Europeans. So Dona Ana de Cinchon, wife of the Viceroy of Peru in 1638, named that tree because there was a preparation of the bark that became common in Peru. And you would take the bark, right? And the bark is rich in quinine. And quinine kills insect herbivores. Right? If you take that bark and you soak it in water, you get a quinine tonic. And if you drink this extremely bitter quinine tonic water, well, it kills malaria plasmodiums. So malaria, as it turns out, is a huge problem absolutely everywhere along the equator. So South America as well. Florida, the Gulf Coast used to have big malaria issues. So malaria all over, anywhere there's an equator. And as it turns out, the cinchona tree, when its bark is soaked in water, you get this quinine tonic that when you drink it, it gets into your blood and kills the malaria plasmodium. And it gives you resistance to malaria, and it helps treat malaria. So you can drink a quinine tonic preventatively all throughout the time that you're in a malaria-ridden place. And if you get malaria, you can drink your quinine tonic. So it is anti-malarial, and it is bitter. It is a bitter tonic. And as it turns out, if you want to mask that bitter flavor, you would often get some gin. And the drink, the gin and tonic, was literally adding gin to your quinine tonic in order to make it more palatable. So when you're drinking a gin and tonic now, the tonic is water that's, supposed to be around as bitter as quinine tonic. So if you go to a bar and order a gin and tonic, an 1800s British soldier would probably ask you why you're voluntarily drinking something as bitter as a quinine tonic. So we can, of course, make quinine synthetically now, which is pretty cool. And the synthetic drug is more effective against malaria than the bark-soaked tonic. So that's kind of cool. Of course, being so effective against the malaria plasmodium means you have an evolutionary pressure on the malaria plasmodium. And so the malaria plasmodium has become resistant to quinine. Malaria plasmodium evolved quinine resistance. So that's kind of fun. We're getting super duper close to the end, folks. 37 of 39. Here's the animal symbiosis in angiosperms. The ants and the acacia tree. The acacia tree produces some pretty gnarly thorns. Acacia thorns are very large. And that's because in Africa there happen to be very large herbivores. Very large for large herbivores. Not that it stops many of those large herbivores. Anyway, they're large enough that ant colonies can live in hollowed out acacia thorns. So acacia ants live in hollowed thorns. That live in, that part there got pretty, anyway. So what do these ants? These ants get a home, right? They get a home It's a pretty spiky, gnarly home, which is pretty cool. Very, very awesome for the ants. Very metal. In addition, right, they feed on acacia nectar. So not all acacia. Like acacia is a family of trees, and there's acacia ants all over, each co-evolved with their various acacia trees. So they get food from the nectar, right? Um, and they attack herbivores, insect, birds, and even mammals. All. Like all ants, they have a critical inability to understand their size compared to other organisms. So they attack insects, they attack mammals, they attack birds, they attack epiphytic plants. Plants that just want to chill on the bark of the acacia tree. They attack those. They keep an acacia tree clear, and they get food from the nectar. Fun fact, the flowers secrete a repellent. They secrete an ant repellent. And why do they do that? Because though the ants co-evolved this really beneficial relationship with the acacia, the ants live on one acacia tree, on the branches of one acacia tree, right? They are crap if you're coming to pollination. So you have to repel your vicious defenders to allow pollinators to feed from the flowers. So that allows the pollinators to feed. Wow, this is a bad color. I don't think there's any good color for this combo. But it allows pollinators to feed and pollinate without getting attacked. Fascinating. In some acacias, the nectar, the acacia nectar that they get inside the acacia tree, is habit forming. In some acacia trees, for some, it's addictive. And for some ants and acacia trees, it's downright symbiotic. And by that I mean there are some ants that can't live without their acacia host. So some of these, there's a species of acacia ant, at least one, that can't digest sucrose. The most common, so one of the major sugars in acacia nectar. They lack the enzyme invertase. And as it turns out, the acacia tree that they live on, its nectar contains invertase. So that's true, like symbiosis, to the point where those ants couldn't survive without their acacia host. So there's like a whole gradient of cool ant-acacia interactions here. So that's pretty cool. Slide 38. This is kind of a fun one, involving parasitoid wasps. I love parasitoid wasps for a number of reasons. There's all sorts of parasitoid wasps out there. Their life cycle is gnarly. It is metal as heck. And they are literally the only species out there that caused Charles Darwin in his writings to question the existence of a loving God. So that's how freaky parasitoid wasps are. See, parasitoid wasps, they often parasitize caterpillars. That's like one of the favored hosts. There are parasitoid wasps for virtually any and every insect. But caterpillars are a big target, and what they do is they lay their eggs sort of in and on the skin of their host. And then when the eggs hatch, the wasp larvae, chew their way inside to the live caterpillar. And the live caterpillar goes into like this hyper feeding mode. So it just feeds nonstop and cranks out as much nutrients as possible, but it doesn't grow because, you know, it's slowly being eaten alive. Not enough that it's a lethal level of getting eaten alive because insects are wacky. They spend a lot of time growing. And then eventually they bust out of the breathing pores on the sides of the caterpillar and the caterpillar dies. So it's some real like alien movie in real life stuff right there. So it's pretty wild. And a cool fact is that when a caterpillar chews a leaf, the leaf releases volatile compounds. Volatile compounds evaporate quickly and diffuse in the air. That's kind of what it means to be volatile. So, you know, parasitoid wasps follow the volatile compounds back to the leaves. They're attracted. The wasp is attracted by the volatile organic compounds released when the caterpillar eats. And that allows them to find a host. So that's kind of cool. The plant's defensive chemical isn't harming the caterpillar, it's attracting a wasp that kills the caterpillar. Kills it a lot quick. Even though the caterpillar goes into hyper munch mode, it still dies a lot faster and sooner than it would have. So it is a net protection against herbivory there, which is wild. Instead of releasing a chemical that kills or harms a caterpillar, which we see, you know, like in milkweeds, you can have caterpillars evolve to protect from that. You go another route, which is attract their predator. And in this case, not even their predator, their parasitic wasp. Oh man. So, yeah. The larvae feed on the host, and then they kill the host caterpillar, and that protects against herbivory. So, then the larvae pupate into adult wasps, and you have a new life cycle going on. So that's wild. I love that. That's metal right there. You're not producing something as lame as a cardiac glycoside or a cyanogenic glycoside. You're attracting a parasitic wasp to come lay eggs in the caterpillar that will then devour it from the inside out. Oh, oof. Right, last slide. Let's talk about a defensive systemic response. So, what is a systemic response? All right, so it is energetically costly to constantly produce your defenses. your secondary metabolite, your secondary defensive chemicals. Right? It costs a lot of energy to produce and store and keep up your supply of cardiac glycosides, cyanogenic glycosides, nicotine, caffeine, all of this stuff. It takes a lot of energy to have that on hand so that anytime an herbivore attacks, you have a defense. So here is kind of a cool alternative energy saver. And that's the systemic response. So the systemic response is when you have a chemical that, so how do I want to phrase this real quick? The systemic response, right? Instead of you do not have a constant supply of defensive chemicals. Right? You're not constantly storing. In this case, jasmonic acid in the tomato. So jasmonic acid binds to digestive enzymes and inhibits protein production or digestion. Inhibits protein digestion. Why have jasmonic acid on hand always when instead you could have a systemic response? It saves energy. Because basically how a systemic response works is you have damage causes the release of cysteamine. So cysteamine is released into the phloem. Right? And that causes a pathway, so it activates the jasmonic acid production pathway. Right? And then that proliferates in the plant. Short term. So, in quick order after damage, you fill your plant leaves up with jasmonic acid. And now, jasmonic acid is inhibiting, so it inhibits protein digestion. Proteinase, ASE means enzyme, protein means it digests protein. So it's a proteinase inhibitor. So the proteinase inhibitor is jasmonic acid, and that's pretty cool. So it inhibits the digestion of the plant. So it's a defense on demand. Much less costly. A lot more complicated than just cranking out cyanogenic glycosides. And there's a delay in the amount you have to, you're restricted by the amount of time it takes jasmonic acid to build up into the plant to a defensive level. So that's kind of cool. Jasmonic acid is the one in tomatoes. So that's kind of cool. In willow trees, in the white willow, salicylic acid. Salicylic acid, which is aspirin, is part of their systemic pathway. So it signals proteinase inhibitors. So basically, jasmonic acid in tomatoes, salicylic acid in white willows. So it's part of a systemic response. So there you go. You don't have to put all your resources into having your defenses constantly in your plant. Instead, you can have them on demand. So the bonus is significantly lowered energy costs associated with defense. The con would be that it takes time. So, you know, I don't know. Pretty cool. It's just literally pro and con, like a cost benefit thing as to whether or not a systemic response is warranted in these various species. So tomatoes, as it turns out, that systemic response is great. Tomatoes also have, various other chemicals in them as well. So, I don't know. Very cool stuff. And this is the last slide as well. So, dear God, this one was 2 hours and the other one was an hour. This is like a 3-hour lecture. I'm sorry. It's midnight. I love you. Good night.